Good morning, Year 12. So, what we're going to do today is we're going to continue with these little catch-up PowerPoints to explain the more difficult topics of biology. And we're going to be doing translation today. Now, translation is the process where we take a piece of mRNA and we use it to create a polypeptide chain, a chain of amino acids with peptide bonds between them. So, very broadly, there's kind of three things we need to be able to do. The first is to describe some features of the genetic code. The second is to describe the process of translation, and that's by far the hardest bit. It's the bit people tend to struggle on. And the third thing is to describe how ATP is used in translation, which actually is a relatively common question, but it's only ever worth 1 or 2 marks. Fine. Let's start off, before we get onto the actual process of translation, let's start off by talking about the structure of the genetic code. So, there are 5 features of the genetic code I'd expect you to know. The first is that it is universal. Now, you could be asked in a test, what does the word universal mean in relation to the genetic code? And what it means is that every single organism on Earth that we're aware of uses exactly the same code. So if you say I have TTT on your first codon, on your first 3 bases of your DNA, that will code for phenylalanine. So you can see, top left of this little table, TTT is phenylalanine. And that's true whether you're a bacteria, whether you're an insect, whether you're a tree, whether you're a human. It's always true. Fine. No, going on. Fine. Non-overlapping. Move my head there. Non-overlapping. Each base is only part of one codon, by which I mean, let's say the code was AAA-TTT. Okay, then AAA would be, let's find it, dear me. There it is, lysine, and TTT would be phenylalanine. It doesn't go AAA, then AAT, then ATT, then TTT. It's 3 bases and then another 3 bases, and you can't have a base that's in the first codon and the second codon. They're separate from each other. Each 3 bases are kind of a separate unit, right? Non-overlapping. So based on this unit, can't be in this unit, right? They're non-overlapping. Probably the hardest one to word, but if you just remember it means that each base is only part of one codon, or one triplet, you'll probably be alright. And I suppose we should come to what the word codon means. I think most people are aware of that term, but let's just make sure. A codon is 3 bases on a piece of mRNA, which code for a single amino acid. We'll come back to that later. Degenerate. Degenerate, each amino acid can have more than one codon. So for instance on leucine here, leucine could be CTT, CTC, CTA or CTG. That means the code is degenerate. It would not be degenerate if each amino acid only had one codon, but it doesn't. Each amino acid can have more than one. Most do. The start code is ATG. Now to be honest with you, that's technically on the A level, but it never gets asked about. ATG is methionine. Methionine is always the first amino acid in every single protein in your body, but it's not particularly relevant and it doesn't come up very often. Stop codes, and this is something that does come up, the idea of a stop code, you can see here, the red ones, TGA, TAA, TAG, they're all stop codes, and a stop code, if a ribosome comes to those 3 bases, that codon, instead of just keeping going, it's a signal to the ribosome where it makes it drop off. The ribosome drops off of the mRNA, it detaches. And so a stop codon is an area where the ribosome detaches from the mRNA. Now we need to know those 6, unfortunately, and the kind of questions you'll see on these will just be, what's universal mean? What's degenerate mean? What's a stop codon? So you've got to know those 6. Now if it's just a question of memorizing it, then do that. Right, now let's talk about the actual process of translation. You start off with a piece of mRNA which leaves the nucleus through a nuclear pore. If you like to take notes here, you're welcome to, right? But you have to pause me as we go along. So it's going to leave the nucleus through the nuclear pore into the cytoplasm. So now we're going to think, well, it's going to get attached to a ribosome. Where are ribosomes found? Ribosomes are found in 2 places, mainly on the rough endoplasmic reticulum, but also free-floating in the cytoplasm. So in one of those 2 places, you're going to have a ribosome that's going to attach onto this mRNA. And we can see that on this piece of mRNA there are codons. In fact, in this particular place there are 7 codons. Now in reality there's going to be hundreds more than that. This is a small piece of mRNA, but that's fine. So we've got a codon here. Now these codons are 3 bases and they each code for 1 amino acid. Now, a piece of tRNA is going to come along and it has to have a complementary anticodon. So we've got the codon is the 3 bases here. On the top, if you like, of the tRNA is an anticodon. Now there are tRNAs floating around all the time. They're just buzzing around the area and they are waiting for their particular complementary base sequence, complementary codon, to be there. That will attract them in and they'll form hydrogen bonds between these A's and these U's. So light comes and it forms hydrogen bonds. Now hydrogen bonds are very weak, they're very easily broken. There's only 3 here, so you could easily rip that back off again, right? Whenever you dip your hand into a pool of water, you're breaking millions of hydrogen bonds. This is just a very weak attraction. But it is an attraction. So now I've got a tRNA kind of hanging on, if you like, to the mRNA. And the ribosome acts as it's not a catalyst, let's be clear, but it almost acts as a catalyst of this reaction. It makes it easier for the tRNA to bind, it gives it a binding site, it holds the tRNA in place. Right, so the anticodon binds to the first codon by weak hydrogen bonds, and then along comes, where's that not working? There we go. Another tRNA with a second anticodon, which is complementary to the second codon. And you can see at the bottom here, AA1 and AA2. AA1 is the first amino acid, AA2 is the second amino acid. And which amino acids are there would depend on this anticodon. Do you remember we said AAA? Do you remember we said AAA would code for lysine? Sorry, TTT of course. Remember we said that would code for phenylalanine, so this would be phenylalanine. I don't know off the top of my head, I've never needed to memorize them since university, but CGA will code for a different amino acid. So GCU, this anticodon, will be a different amino acid. The amino acid that's attached is based on the anticodon. Now, in a minute, the ribosome is going to move along. And if you're not careful, the tRNA will just drop off, taking its amino acid with it. So we need to bind this amino acid onto the other amino acid. So we form a little peptide bond. And the peptide bonds are incredibly strong compared to hydrogen bonds. And so the peptide bond is going to be much stronger than this hydrogen bond. And in fact, it's going to be stronger than this little black bond here. And so this amino acid is fairly much glued to this other amino acid. So we're forming a peptide bond. Now there are enzymes involved here, it's actually a lot more complicated than that, but at A level, we're happy just to say that a peptide bond is formed. Then, the ribosome moves along, and the tRNA drops off. So we've got the ribosome moving along, the third tRNA comes along with its anticodon, and it's got complementary to the codon, it's got a third amino acid. Now what's going to happen now? I'm sure you can guess, we get a peptide bond. So we just keep going like that. And along moves the ribosome, and every time you've got a tRNA attaching and binding to the previous amino acid. Now you come to CGC. Now this is a stop codon. Now that means that there's nothing for the ribosome to bind onto, as the ribosome detaches and the polypeptide chain is released. And this is your polypeptide chain. Now that means folding into its secondary, tertiary and quaternary structure, but that is going to be the basis of having a protein made. So, we go through that one more time from the beginning, just very, very, very quickly. 1. The mRNA leaves the nucleus through the nuclear pore, enters the cytoplasm, and it attaches to a ribosome in the RER or the cytoplasm. It's got codons all over it. The tRNA have anticodons, they're complementary. The anticodon binds to the codon. Another anticodon binds to the codon. So now I've got 2 tRNAs with 2 amino acids. They get a peptide bond between them. The ribosome moves along, the tRNA drops off. Third tRNA comes along and binds. Peptide bond. Peptide bond. Peptide bond. Peptide bond. And the end. Stop codon's reached. The ribosome releases the mRNA and the polypeptide chain. Now, several ribosomes can pass along the same piece of mRNA, the first. So this is like, have having a single copy of a recipe for a cake, by having 500 bakers all reading it from around a table, right? You've only got one mRNA, but actually you can make hundreds and hundreds of proteins out of that, right? There's only one recipe, but you can get hundreds of things made, because lots of ribosomes can get involved. Now, that's the process of translation, and it's relatively complicated. The basic ideas are very simple. Go through a question on that in a second. Now, the final thing to understand is what we're using ATP for here. Now, this is a question they love to ask for some reason. ATP is used to bind an amino acid onto a tRNA. That doesn't just happen. tRNAs aren't made with amino acids attached. The amino acids are made in a different process. The tRNA is made by transcription. It's a special kind of it's a special kind of RNA. You have to bind them together, and that takes ATP. You break it down, and you make yourself a nice tRNA with amino acid. So they're activated, if you like, after use by recombining with a specific amino acid using ATP. Fine. Now there's two questions here. And these are the classic story questions. They're the reason we're doing this video, because these questions come up all the time in one form or another. Now I'd like you to have a look at those. There's 6 marks there as usual. And I'd like you to have a go at, without looking back if you can, without looking at a textbook, I'd like you to have a go at these 6 marks. You can bullet point it, particularly if you've been taking notes. There's no need to write it out in full again. If you haven't been taking notes, this might be a good opportunity to write it out in full so that you have the notes there. So pause me, take 6 minutes in this, and try and get all 6 marks based on what I've just said. Okay? Right, now, starting with mRNA, describe the process of translation leading to a production of a polypeptide. So, mRNA attaches to ribosome, 1 mark. Codon on mRNA, 2nd mark. Binds to an anticodon on tRNA, 3rd mark. Each tRNA brings a specific amino acid. That's something people forget to write. It's also something teachers forget to say sometimes. I may not even have said it in this video. I wouldn't lose you all your marks here because there's more than 4 marks, but it's always worth writing. It's quite important if you think about it. If a tRNA doesn't have a specific amino acid, then it's irrelevant what the anticodon is. If a tRNA can have any amino acid, then the anticodon is meaningless because the tRNA will just bring a random amino acid. No, but each tRNA has a specific amino acid. The sequence of codons or bases on the mRNA determines the order of amino acids. An obvious point, but one that's worth writing. And you've got the formation of peptide bonds or amino acids joined by condensation reactions, which is the same thing because you form peptide bonds by condensation reactions. Now, the marks people miss out of these 2 down here, as I've said, you can still get 1, 2, 3, 4 marks without using these 2 in the middle. It only comes difficulty when it's a 6-mark question, which it sometimes is. So memorize this, get it in your head, write down anything you didn't write down, and learn it. Right, learn it. If you haven't learnt it, just practice it over and over again. Test yourself on it occasionally. Even more often coming up is this meaning of degenerate, non-overlapping, universal, etc. Let's have a look. So degenerate, more than 1 base or triplet for each... No, more than 1 triplet for each amino acid. If you said more than 1 codon for each amino acid, that's fantastic. They just didn't want to take your mark away for not using the word codon. Codon is a better word than triplet. So more than 1 codon for each amino acid, you'd accept more than 1 triplet for each amino acid. Non-overlapping, each base is part of only 1 triplet or codon. Fine. That's the end of that. Now if you've got all of that, if you've got 6 marks there, then that's translation. There is nothing else, what we've just done. If you didn't get those 6 marks, let's say you didn't remember degenerate and non-overlapping, go back, either watch the video again if you think it'll be helpful to you, or go and learn those from a textbook. Literally sit down at the end of an evening, a couple of nights every now and then, and just go, right, I need to memorize degenerate, non-overlapping, what do they mean? Oh yeah, I forgot that wrong, I'm going to practice again tomorrow. Until it's stuck in your head, because there's no good way to remember it except to memorize it. Right, that's the end of that. I hope it's been helpful to you. If there's anything you didn't get there, feel free to contact your teacher and I'm sure they'll be able to explain it to you. Have a great day.